Welcome back, class, to the subject Applied Genomics and Plant Breeding. Tonight we're going to take a closer look at the genetic control of the development of the body plan of an organism. So let's start by imagining the formation of a complex animal or a complex plant from a single-celled egg. This involves an amazing transformation that organises that massive cells into a distinct form with a head and tail, or a shoot system and root system, appendages in the case of an animal, tissues, organs and systems. But biologists were stumped for many decades regarding how this biological form is generated during development. And the key catalysts to understanding how these complex forms were developed were the discoveries of genetic monsters, mutant fruit flies. These mutants had dramatic alterations of body structure, and in the early days of studying development or studying embryology, the favourite genetic model organism was the fruit fly, called Drosophila melanogaster. So these mutants arose as a result of spontaneous, rare mutations, or simply as byproducts of other experiments. So let's go back to 1915, when a fly was isolated with a mutation that caused the tiny hind wings, called halteres, to resemble the large forewings. And the chap that discovered this called this mutant bithorax. This transformation was referred to as being homeotic. Homeotic comes from the Greek homeos, meaning same or similar. And this type of transformation was called homeotic because one part of the body was transformed to resemble another. Then additional mutants were identified in Drosophila, the fruit fly. For example, the Antennapedia mutant, and we'll come back to that one in just a minute. So homeotic mutants inspired this revolution in the study of development and embryology. And basically, especially once the tools of the molecular biologist became available. So in this lecture, we're going to look at a few concepts that illustrates the logic of genetic control of the development of the body plant. And what was surprising also, I should mention, was that these strange fruit fly mutants and the genes that were causing the mutations turned out to be the key to the study of the entire animal and plant kingdom, because counterparts to these genes were discovered that played similar roles in almost all other animals and plants that were studied. So here we can see some of these bizarre mutants of Drosophila melanogaster, the little fruit fly. So in A, on the right-hand side at the top, we have a normal fruit fly, and we refer to a normal plant or animal as a wild type. So this is a wild type fly. It has one large pair of forewings on the second thoracic segment and one very small pair of hind wings, which are really difficult to see on the third thoracic segment. Then just under that in B, we can see a mutant. And this mutant is referred to as a triple mutant because there are 3 mutations in a gene called the ultra bithorax gene. And these three mutations cause the development of forewings, another pair of large forewings in place of those very small hind wings. Then at the bottom in C, we can see another mutant called the Antennapedia mutant. And if we look at what should be the antennae of this fly, they've actually been transformed into legs. Even more stunning is the images to the left, which shows the small pair of antennae in the top image, and then at the bottom we can see that there are legs in place of where the antennae should be, and that's indicated there. So quite bizarre mutants that really sparked this field of study. So for many decades, the study of embryology and development largely involved the physical manipulation of embryos and cells and tissues, for example, through experiments called transplantation experiments, which we'll look at on the following slide. And these transplanted regions of cells, for example, were called organisers because they had the ability to organise the development and affect the development of surrounding tissues. Now, these organisers were postulated to produce morphogens, that is, molecules that can induce very different responses in the surrounding cells or surrounding tissue in a concentration-dependent manner. And although this discovery was quite spectacular, progress in understanding the nature of these organisers and morphogens stalled after the discovery in the early 1900s. And the reason that progress stalled was because it was impossible at that time to isolate the molecules that were responsible. So here we have some organisers, examples of organisers that were discovered in animal embryos. So if we look at A, just the image on the left-hand side, you can see labelled there by that arrow a region of an embryo, part of a developing amphibian embryo. This region was transplanted to another site in a recipient embryo, which is shown in the middle on that top panel. And that organiser, that transplanted organiser region, was shown to induce the surrounding tissue to form a second complete body axis, which we can see at the top right-hand side. We've got another example here in B. So a region labelled here as ZPA was transplanted, and this was transplanted from the posterior part of a developing chick bud limb to the anterior part of the chick limb bud. And this organiser could induce extra digits, but those digits had reversed polarity with respect to the normal digits, which we can see to the right in B. The breakthroughs came around with the age of genetics. That is mainly the isolation of mutants with very discrete defects in development, and the characterisation and study of genes and gene products. So the genetic approach to studying development and embryology provided many advantages. 3 are listed here. So firstly, assumptions were not required about the number or nature of molecules involved in a process. Number two, the limited quantity of a gene product, whether that's messenger RNA or protein, was no impediment. All genes can be mutated regardless of the amount of product they make. And number three, the genetic approach can uncover phenomena for which there is no other assay. So from a genetic viewpoint, there are key questions that are concerned with the number, identity, and the function of genes that are involved in development. And here are some key questions listed here. So number one, which genes are important in development? Number two, where are these genes active? That is, in what cells, what tissues, and at what times are they active? Number three, how is the expression of developmental genes regulated? And number four, what are the molecular mechanisms that cause these gene products to have an effect on development? One of the first considerations in genetic analysis was which animal or which plant to study. The fruit fly, Drosophila melanogaster, emerged as the leading genetic model of animal development. And it was the favourite due to its ease of rearing, its very rapid life cycle, its cytogenetics, that is the all the details about its chromosomes, and decades of classical genetic analysis. The nematode worm, Caenorhabditis elegans, was also commonly used. The lab mouse was used for vertebrates, for study of vertebrates. And a more recent model organism, the zebrafish, was a favourite, became a favourite because the embryo was transparent. What about the model plant? Well, there were several different plant species that were commonly studied, maize, tobacco, for example, but eventually, probably around the 1980s, the model plant Arabidopsis thaliana became the number one favourite. We're going to focus in this lecture on what's called the genetic toolkit for the fruit fly, Drosophila, because its identification was really the major source of insights into the genetic control of development. So animal genomes typically contain about 13,000 to 20,000 genes, many of which function in essential processes in all cells. So, for example, genes that are required for cell respiration, genes required for the biosynthesis of macromolecules, such as proteins and lipids and carbohydrates, and these genes are called housekeeping genes. Other genes are more specialised. Here they were interested in a different set of genes that determine the overall body plan, and also the number, identity, and pattern of body parts. And these genes were referred to as toolkit genes. So several hundred of these toolkit genes have been identified so far out of a total of about 14,000 genes in the fly genome. So many gene mutations are lethal when they're hemizygous or homozygous because the cells simply cannot survive without the products of these genes that are affected by these mutations. So they needed mutations that cause some very discrete defect in the embryo or in the adult body pattern or both. So these toolkit genes became classified according to their function in controlling the identity of body parts. So, for example, controlling different segments or appendages, genes involved in the formation of body parts themselves, or involved in the number of body parts, in the formation of certain cell types, or in the organisation of the primary body axes, that is the AP and DV axes, which we'll come back to. So among the most fascinating of transformations are the homeotic transformations, and many of these have been observed in nature, and we'll see some examples on the next slide. A mutation in a gene may cause a loss of gene function or a gain of function. So for example, a loss of function mutation in the ultrabithorax gene results in repression of forewing development. Gain of function mutations, on the other hand, in the ultrabithorax gene transforms the forewing into a hindwing. And another example, a gain of function mutation in the Antennopediae mutants causes the transformation from the antennae to legs. Here we can see homeotic transformations, and these are drawings that were done in the late 19th century. So in A, on the left of screen, we can see a fly. This is not Drosophila, this is a sawfly, and the left antenna has been transformed into a leg. In B, we can see three specimens of the vertebral column of a frog. The one right in the middle, this is normal. The left specimen has extra structures growing out of the top of the vertebral column, and the one on the right has an extra set of vertebrae. Although the homeotic genes were first identified through mutations, they're actually required throughout the life of the fly. Systematic searches for these homeotic genes led eventually to the identification of 8 loci, now referred to as Hox genes. And these Hox genes affect the identity of segments and appendages. Complete loss of Hox gene function results in embryo lethality. But the mutations in adults are viable in heterozygotes because normal gene function is provided by the wild type allele. And that's quite common to have a mutation in one allele which doesn't result in any change in phenotype, because the product of the normal allele on the other chromosome provides enough of the required product. So they found an intriguing feature of the Hox genes, and that was that they're clustered together in 2 gene complexes found on chromosome number 3. The one complex was called the bithorax complex, and it consisted of 3 Hox genes. The other complex was named the Antennopedia complex, and there are 5 Hox genes in this complex. And also intriguing was that they found that the order of the genes actually corresponded to the order of the body regions in the fly that are actually influenced by each gene. And we'll see that on the next slide. So the relationship between the structure of the Hox genes and the phenotypes of the mutants was actually revealed when they were able to characterize the molecular basis of those genes so as to then prove that relationship between the two. So, for example, you need to show that a gene is expressed in the region where the mutant phenotype is seen. So technologies for enabling visualization of gene expression in embryos and any other tissues in any organism are, number one, to look at the expression of the RNA transcripts, and these can be visualized by using a technique called in situ hybridization. Another technique involves looking at the expression of proteins, and this is done using immunological methods. Both of these methods require cloning of what's called cDNA, where the cDNA is referred to as complementary DNA or copied DNA. So here we have at the bottom of the screen the 8 Hox genes found in 2 complexes on chromosome number 3. Each one is given a different colour, and that colour coding identifies the segments and the structures in the fly pictured here that are affected by mutations in each gene. This slide illustrates the 2 main methods used for visualizing gene expression in developing cells, tissues in both animal and plant. So in the embryo, they found that Hox genes are expressed in spatially restricted, sometimes overlapping regions. The patterns of expression of these Hox genes correlated with regions of the animal that were affected by mutations in those genes. So, for example, in the image that you can see here, the purple shading indicates where the ultra bithorax gene is expressed in the posterior thoracic and most of the abdominal segments. The actual expression is seen as different colours of each one representing one of the Hox genes. And this was actually shown by using in situ hybridization. The different genes that are shown here by the various colours have quite unusual names. One is called deformed, one is called sex chromes reduced, then we've got antennopediae, ultrabithorax, abdominal A and abdominal B. So the Hox genes were found to control the identity of the structure, but not the formation of the structure. So for example, limbs will form, but they all have antennal identity. Another example, wings will form, but they all have forewing identity. An important question that was asked was, what of the nature and structure of proteins encoded by the Hox genes? They noticed that the multiple loci had actually arisen by duplication of an ancestral gene. And this led researchers to search for similarities in the DNA sequences of all the Hox genes. And of course, what they found was that all 8 Hox genes in the 2 complexes were in fact very similar, particularly in a 180 base pair region. They named this the homeobox, and the protein domain of 60 amino acids that's encoded by that homeobox was called the homeodomain. In this image at the bottom of screen, we can see the sequences of the fly homeodomains. So these are the sequences of the 8 Hox proteins, but just the homeodomain regions. And all were found to contain a very highly conserved homeodomain composed of 3 alpha helices. And you can see that that's labelled there right at the bottom of the screen as helix 1, helix 2, and helix 3. So this was found to form what they call a helix turn helix motif, which is very similar in structure to yeast mating type proteins. This suggested that Hox proteins are sequence-specific DNA binding proteins. That is, they're proteins that bind to regulatory elements of other genes to either activate their expression or repress their expression. And this class of DNA binding proteins is generally referred to as transcription factors. So transcription factors are proteins which will bind to promoter regions, for example, of other genes and either switch them on or switch them off. And many of the toolkit proteins actually turned out to be transcription factors. When the homeobox was discovered in fly Hox genes, it raised the question, are they unique to the fly or are they found in other species? So the hunt was on, and they very, very quickly found these Hox genes in other insects, in the earthworm, frog, cow, human, mouse, and many, many others. They also found lots of these Hox genes in plants. There's actually about 100 in plants, quite a lot of them in plants, and they're called MADS box proteins. MADS, MADS box proteins in plants. The sequence similarity between these Hox genes across completely different species was astounding because of the similarity of the DNA sequence. In fact, they were nearly identical. They've reported sequence similarity between these genes to be as high as 59 out of 60 amino acids. That's quite amazing. And this suggests that a Hox complex existed in a distant common ancestor more than 500 million years ago, which was when they last shared a common ancestor. So Hox genes must play a fundamental role in the development of most animals and also many higher plants. So why would such different organisms have these sets of genes in common? The role is apparent in that Hox gene expression occurs in overlapping domains along the antero-posterior axis, the AP axis, which we can see in this fly at the top left of screen. So antero-posterior axis is referring to the head to tail axis. They also found that Hox genes also specify the identity of serially repeated units, such as the transformation of lumbar vertebrae to sacral vertebrae in the mouse. Then in the late 70s and 80s, a group of researchers from the famous Max Planck Institute in Germany started the search for genes that are required for the formation of body parts. But they realised that genes are probably embryo lethal in homozygous mutants. So they came up with a screen to search for genes that were required just in the egg before the zygote genome is active. And these genes, were looking for genes that are required for proper patterning in the embryo. Genes provided by the female to the egg were referred to as maternal effect genes, and they depend only on the genotype of the female. They discovered genes required for the proper number and pattern of larval segments, genes required to make the 3 tissue layers, and genes required to pattern the fine details of the animal's anatomy. So the 3 tissue layers that they were looking at were the ectoderm, mesoderm, and endoderm. They did this by using chemically induced mutations. That is, they would saturate the fly's chromosomes with these chemically induced mutations so that they could identify most of the genes required for the building of the fly. The most common chemical mutagen that was used was called EMS, ethyl methane sulfonate. And they were awarded the 1995 Nobel Prize in Physiology or Medicine for their discoveries. Only a few dozen genes were found to be required for the proper organisation of the antero-posterior body axis of the fly embryo. And these genes were grouped into 5 classes on the basis of their influence on embryonic patterning. Number 1, genes that set up the AP axis and consist of maternal effect genes. So, for example, the bicoid gene. Embryos that have a mutation in the bicoid gene were missing the entire anterior region. The second class of genes included the gap genes, and gap genes affect the formation of a whole block of segments within the embryo. So, for example, mutations lead to large gaps in segmentation. Let's look at those on the next slide. So on the left, we can see the results of a mutation in the bicoid gene. This is one of the maternal effect genes, and the bicoid mutant is actually missing the anterior or head region. Then they also discovered what are called gap genes. And we can see the result of mutations in the gap genes. These lead to large gaps in segmentation. And 2 genes shown here are the Kruppel and NURPS gene. Then in the center of that image, we have what were called the pair rule genes. One is named even skipped and one is called odd skipped. And then we've got 2 more at the bottom called paired and runt. On the right, we've got 2 examples of segment polarity genes, gooseberry and patched. So we'll talk about some of those classes on the next slide. Here we have number 3, class number 3, the pair rule genes. These affect pairs of segments. So mutants are missing part of each pair of a segment. So for example, we saw the even skipped and odd skipped gene. Class number 4 include the segment polarity genes, which affect patterning within each segment. Mutants display defects in segment polarity and number. And class number 5 the Hox genes, which do not affect segment formation, but alter the appearance of the segments. So in order to understand that relationship between the genes and the phenotypes seen in the mutants, we need to know the timing of gene expression and the location of gene expression. So for example, the maternal effect bicoid protein was found to be expressed in a graded pattern from the anterior pole of the embryo. And this just happened to be the section that was missing in the bicoid mutant. So in A, at the top of the screen, we can see the bicoid maternal effect protein, which is expressed in a graded pattern from the anterior pole. So you can see that darker shading to the left of that embryo, which represents the bicoid protein. And of course, this is the end of the embryo that's missing in the bicoid mutant, as we said on the previous slide. In B, they could see the expression of the Kruppel protein, which is a gap protein, and it seems to be expressed in a large segment in the center of the embryo. In C, they're looking at the pair rule protein called hairy, and hairy is expressed in every second segment for a total of 7 stripes, which covers the 14 future body segments. And then in D, we have a segment polarity protein called engrailed. And engrailed is expressed in groups of cells within each segment. So they showed from this that the protein expression in the embryo, that is, it correlates with the phenotype that they saw in the mutant. So on the previous slide, we saw the maternal effect bicoid protein being expressed in quite a large region in that graded pattern from the anterior pole of the embryo. We also saw the gap protein Kruppel, which was expressed in a very large region of cells corresponding to a future position of a segment. And then we saw also an example of a pair rule protein, for example, hairy, which is expressed in striped patterns. So note the progressive refinement in gene expression as development proceeds. The first genes are expressed in very large regions in the embryo. These are the gap genes. Then we've got genes that are expressed in stripes, 3 to 4 cell wide. So for example, the pair rule genes, such as hairy. And then we have expression, even more refined expression from the segment polarity genes, such as engrailed in stripes within the embryo, consisting of 1 to 2 cells in width. So it was found that the order of gene expression was quite logical. First, the maternal effect protein bicoid appears. That's followed by the zygotic gap proteins, then the pair rule proteins, and lastly, the segment polarity proteins. This order of gene expression and the progressive refinement in terms of where the genes are expressed revealed a step-by-step process. And this also suggested that one set of genes may actually govern the expression of a succeeding set of genes. In this table, we've got some examples of Drosophila AP axis genes that contribute to pattern formation. So we can see the gene names here: hunchback zygotic, crupple, nerps, even skipped, Fushitarazu, odd-paired, paired, engrailed, wingless, hedgehog, patched, labial, deformed, antennapedia, and ultra-bithorax. In the next column, protein function is indicated, and you can see there that most of these, the proteins encoded by these genes are actually transcription factors, with a couple of signaling proteins in there. Their roles in early development are listed in the final column as either gap genes, pair rule genes, or segment polarity or segment identity genes. The dorsoventral axis is also divided into regions, and they found several dorsoventral maternal effect genes. Keep in mind that dorsal genes are required to establish regions in the embryo from dorsal, that is top, to ventral, bottom. Or you can think of it as from the back to the front. Dorsal mutants lacked ventral structures such as mesoderm and the nervous system. The dorsal protein was found to be expressed in a gradient along the dorsoventral axis, with highest levels in the ventral cells. Each region was found to have different levels of the concentration of these proteins, and different zygotic genes are expressed in each region to affect the patterning of the embryo. In the image on the left, we can see the maternally derived dorsal protein expressed in a gradient with the highest concentration in the ventral cells. On the right, we can see the different colours within the embryo, which represent expression of 4 different zygotic dorsoventral patterning genes. So one gene shown in yellow, one in green, one in red, one in green, and one in blue. So how do the genes know when and where to turn on and off in these precise patterns? How does each cell know where it is in space with regard to its position in the embryo? In other words, how are the spatial coordinates of the developing embryo conveyed to the genes? As we said earlier, gene expression is controlled in many cases by transcription factors, and these transcription factors act on regulatory elements in the DNA of other genes, so they're referred to as cis-acting regulatory elements. These cis-acting regulatory elements may be in the promoter of the gene, or they may be even further upstream of the gene itself. So in order to define a position in an embryo, information must exist that distinguishes that position from others. This positional information indicates longitude, that is the location along the AP axis, latitude, which is the location along the DV axis, and altitude or depth, that is the position within the germ layer of the embryo. So we're going to look at a few examples of how genes are regulated to be expressed spatially, that is within a specific location. Let's start with the bicoid protein. So this is a homeodomain transcription factor. It's translated in the egg and it's localized at the anterior pole. It diffuses through the cytoplasm. And keep in mind that in the early Drosophila embryo, this embryo actually lacks any cell membranes that would impede the diffusion of proteins. So bicoid can actually diffuse through the entire embryo. So the bicoid protein diffuses through the cytoplasm, and thereby establishing a protein concentration gradient. It remains highly concentrated at the anterior end, and this concentration gradient of the protein provides the positional information. High concentration indicates the anterior part of the embryo. A low concentration of the protein indicates the middle of the embryo. No protein at all indicates the posterior region of the embryo. So let's look at the image in A on the left-hand side. And this illustrates the concentration gradient of the bicoid protein in blue. There's very little bicoid protein present beyond the middle of the embryo. But it's certainly, you can see that it's the concentration is high to the left of that embryo. In B, we can see that the bicoid protein, in fact, 3 bicoid proteins are actually binding to 3 different sites just near the promoter region of a gene. And this gene that's being investigated is called the hunchback gene. But what they've done, they've actually removed the gene itself and replaced it with a reporter gene. And that reporter gene enables you to visualize where the original gene would normally be expressed. And that's because that expression is being driven still by the regulatory bicoid proteins. So that's in the first image in B. And the reporter gene is showing expression indicated as that green expression in the embryo to the right. And we can see the establishment of a protein gradient. Then in the image just below that, what they did was that they deleted one of those bicoid protein binding sites, so that now only 2 proteins bind rather than 3. And to the right, you can see the expression has been restricted. That is the expression, normal expression of the hunchback gene. And then below that, they've deleted another one of the binding sites, so only 1 bicoid protein can bind, and this restricts the hunchback expression even further. Right at the bottom, you can see that all 3 binding sites have been deleted, and now there's no hunchback expression in the embryo. So it was found that several of the zygotic genes, including the gap genes, are regulated by these gradients of a bicoid. So for example, the hunchback gene that we saw on the previous slide is activated in the zygote in the anterior half of the embryo. And we saw that the bicoid protein binds to 3 sites just upstream or 5 prime, you can say, of the hunchback promoter. Activation of that hunchback gene is due to concentration gradients, and these were seen by performing tests using reporter genes. And we also saw that on the previous slide. Here we have two commonly used reporter genes, the LAC-Z reporter gene or the GFP reporter gene. We saw on one of the previous slides the expression of the pair rule gene in 7 stripes within the embryo, and this is actually the first sign of organisation of the embryo. So how are these patterns, these 7 stripes, actually generated? They found that the stripes are patterned one at a time, and the cis-acting elements of individual genes are controlled independently. So, for example, the second stripe is expressed by the gene even skipped, and that lies within the hunchback region. This overlaps with the gap proteins giant and cruple, and even overlaps with bicoid. They found that no other stripe area within the embryo will contain these proteins in these specific proportions. We can see that illustrated on this slide. So if you look at the embryo in image A, one of the stripes is indicated there in blue, and it's called the Eve stripe 2. So let's look at regulation or formation of that stripe by different levels of different proteins. And we can see that in B, in the graph in B, where on the y-axis, this indicates the concentration of the regulators. On the x-axis, this indicates the position along the embryo. So Eve stripe 2 is indicated with that dashed line. Now let's look at the concentration, the levels of the different proteins. So the green line indicates that we have a very high level of the hunchback protein. In blue, a lower level of bicoid, and then lower levels of again of giant protein and the cruple protein. In C, we can see the Eve gene, and also regions called enhancer regions. Look specifically at stripe 2 enhancer region that sits upstream of the actual coding region of the Eve gene. That stripe 2 enhancer region indicated in blue is enlarged in D. And in D, they found that in the enhancer region, there were several binding sites for each transcription factor. That is for the repressors, giant and cruple, and for the activators, bicoid and hunchback. So the net output of this combination of activators and repressors and their relative levels is the expression of the narrow Eve stripe number 2 within the embryo. So they found that formation of stripe number 2 was controlled by a specific cis-acting regulatory element, which they called an enhancer. And this enhancer contained several binding sites for the 4 different proteins. So the entire 7-striped pattern is the sum of different sets of inputs into the different regulatory elements. Bicoid and hunchback activate the eavesstripe 2 element over a broad region. Then you have giant and Kruppel proteins. These are repressors that sharpen the boundaries of the stripe to just a few cells. Therefore, the eavesstripe 2 element acts as a genetic switch. For your interest, the eavesstripe 2 is only 3 to 4 cells wide in the embryo. So how are the different segment identities established? There are 2 aspects to this. One is that Hox genes are expressed in different domains along the antero-posterior axis, and Hox gene expression is controlled by segmentation proteins, especially the gap proteins. 2, regulation of target genes by Hox proteins. So we'll examine one example of how a major feature of the body plan is controlled through integration of many inputs by a single cis element. It was found that paired limbs, mouth parts and antennae each develop from small populations of only about 20 cells. They also observed that different structures develop in different segments in the head and thorax region. The first sign of development of these structures is the activation of genes in very small clusters of cells, which were called the appendage primordia. And this starts with a gene called distallus. Distallus was found to be a key target of the Hox genes. Let's start with A at the top of the screen. Distallus expression as indicated here by the red marks. This indicates the position of the future appendages. In pink, the ultrabithorax gene expression marks the position of the abdominal segments, A1 through to A7. While the gene called engrailed, indicated in blue, marks the posterior of each segment. So distallus is repressed in A1 through to A8. So how is it that distallus is restricted to the anterior segments? They found that distallus expression is repressed by 2 Hox proteins, ultrabithorax and abdominal A, working with 2 segmentation proteins. This was because the cis acting element in distallus contains binding sites for Hox proteins. If these binding sites are mutated, then distallus expression is no longer repressed in the abdomen. This image shows how Hox proteins and segment polarity proteins control appendage location. In A, we have the wild type, that is unmutated cis-acting regulatory elements, with normal expression in the embryo to the right. And we can see that distallus expression is repressed in segments A1 to A7. In B, we have the 2 Hox gene mutations, resulting in de-repression of distallus in segments A1 to A7. Then we have some impacts as a result of mutations of other regulatory elements. So in C, we have a mutation in SLP, the SLP binding site. SLP is the name of another gene called sloppy paired. This is a segment polarity protein. In D, we have a mutation in EN, which is engrailed, another segment polarity protein. In E, mutations in both sloppy paired and engrailed, and some additional mutations in F. These ones are extradentical and homothorax. So they found that other proteins were also involved. For example, the segment polarity proteins are sloppy paired and engrailed. They were expressed in stripes that mark the anterior and posterior of abdominal segments respectively. Each protein also binds to the distalless cis elements. When the binding sites were mutated, then distalless expression is no longer repressed in some of those segments. Two additional proteins were also found to be involved, extradentical and homothorax. They're expressed in every segment, and they also bind to repressed distalless in the abdominal region. So in summary, 2 HOX proteins and 4 other transcription factors bind within a 57 base pair region to repress distalless expression in the abdomen. This is a clear demonstration of how HOX proteins regulate segment identity and the number of body structures. So we have this combinatorial and cooperative regulation of gene transcription, which imposes specificity on the patterns of gene expression.